Welcome, everyone. Today, the Dev Research Seminar is joined with the NISD Thought Leader Seminar Series, and I'm very pleased to introduce Leena Tripathi, who is the Principal Scientist in Plant Biotechnology and the Director of the East Africa Hub at the International Institute for Tropical Agriculture in Tanzania. She is now also joining the NISD as External Advisory Board. Leena gained her PhD in plant and molecular biology and her MSc in molecular biology and biotechnology, and she's worked at the University of North Carolina in Greensboro before joining IITA. She's since been working in plant biotechnology for crop improvement for about 20 years, and she has a special interest in improving banana, plantain, and NSAID, as well as cassava and yams for disease and pest resistance. After our seminar, we'll have plenty of time for questions, and if you could write any questions you have into the chat window, then I will collate them for the Q&A. So Leena, the floor is yours, and I'm just going to switch over to your slides now.
Thanks, Peter, for inviting me, and this is great opportunity for sharing my research with NISD, but all other people who are attending the seminar online. And good afternoon, good evening, good morning, wherever you are, and thanks for the kind introduction. So the topic of my presentation today is navigating science and policy in improving roots, tubers, and bananas through biotechnology. Next slide, please. So let me briefly introduce my institute. So I work at International Institute of Tropical Agriculture, IITA. IITA is one of the 15 CGIAR research centers. It was founded in 1967, and it's headquartered in Ibadan, Nigeria. So the CG centers are actually across globally in different continents. And IITA is one of the world's leading research partners in finding solutions for hunger, malnutrition, and poverty. It has delivered more than 70% of the CGIAR's impact in sub-Saharan Africa. And IITA has been awarded Africa Food Prize in 2018 for research leadership and innovation. Next slide, please. IITA operates with 4 different research hubs, and we have 21 stations in 30 different countries. So we have West Africa hub, Central Africa, East Africa, and the Southern Africa. And as Peter mentioned, actually, I lead the research in the East Africa hub. We work on 8 different crops: banana, plantain, cassava, cowpea, maize, soybean, and yam. And we have about 200 projects and a lot of scientists working at IITA, and most of them are international. Okay, next slide, please. So this is what you see on this slide are the pictures of some of the facilities where I'm based in Kenya, where all the transgenic and genome editing research is based, and that's Becca ILRI hub. Okay, next slide. People going hungry in the world every day, and as you are very much aware that by 2050 there will be estimated 10 billion people, and in order to feed all those people, we need to have agriculture production doubled with the same amount of resources. So the land, water, and all other resources will remain the same, and that's a big challenge, how to double the agriculture within that limited time of period. Next slide. And on top of it, another challenge is the climate change, which is already, we started seeing some negative impact on the crop yield because of the climate change. Next slide. And Africa, if we talk about Africa, actually the population of Africa is projected to be doubled by 2100, Africa will contribute about 82% of total growth, what you can see in the graph here in blue. And so what it means is that we are looking for improving agriculture in Africa. It includes the recognition of farmers' need, that means the inputs what farmers require, high yielding varieties, a widely availability and affordable planting material, which is a key, and access to the market. Next slide. So in order with increasing demand for food and limited resources, we need better and more efficient ways to produce food. And one option is using biotechnology, that either through genetic engineering or maybe more advanced genetic engineering, which is genome editing. So biotechnology is not, even though it has a lot of potential to develop solution for increasing agriculture productivity, but it's not a silver bullet. So in order to enable intensification of crop productivity, we need to use the conventional technologies and the biotechnological application together. And when I say biotechnological application, is not only GM or genomics and tissue culture. Next slide. So this is to show you what's the regulatory status and what's the status of the cultivation of the GM crops globally. So there are 19 biotech mega countries growing GM crops over on about 50,000 hectares of land. And when we come to Africa, actually Nigeria, South Africa, Swaziland, Malawi, Sudan, and Ethiopia have commercialized the GM crops. So, I'm very happy to say that, the several new countries has been added, like, Nigeria, where the cowpea has been approved, Ethiopia, where the BT cotton, and as well as Malawi. So these three countries has been just added last year, who are growing now the GM crops. So why RTB crops? So RTB crops are staple crops, mainly in Africa, playing a major role in food security and income generation. But also they present the common challenges because most of them are vegetatively. So the common challenges are pathogens, pests, and also the slow breeding methods. And as I mentioned that they are vegetatively propagated, means clonally propagated rather than the seeds. So the yield reducing pathogen and pest build up over time. So that's a very big challenge for the RTB crop. And recent advances in the new breeding techniques like genetic engineering, genome editing, have the potential to accelerate the breeding of the RTB crops. So as Peter mentioned that, my group is mainly focusing on bananas. And banana is one of the major staple food crops grown in over 136 countries. And when we talk about Africa, it is the fourth most important crop. And world production is about 153 million tons of banana, and about a third of those bananas are produced in Africa by smallholder farmers. Even though India is the largest producer of banana, and within Africa, East Africa is the largest banana producing as well as the consuming region in Africa. So in Africa, annual consumption in several countries is pretty high. It's about like 21 kilogram of banana plantain per capita. For example, Ugandans consume 191 kilogram per year per person, and which relates to 0.7 kilograms daily per person. And so for them, banana is a staple food similar to the potato in UK. And they eat it with, so it's lunch and dinner for them. But still there is a big yield gap when it comes to the production in banana. So in Matoke varieties, which are the cooking type of banana, so the current production is about 30 tons per hectare per year. But the potential is 70 tons per hectare per year. So you can see the yield gap there. And the similar situation is with the plantain, where the current yield is 7.8 tons per hectare per year, but it has the potential of 35.5 tons per hectare per year. And you might be wondering, what's the reason behind this yield gap? So banana production is constrained by several diseases and pests like so fungal diseases like black sigatoka, fusarium wilt, there is bacterial wilt, also viral diseases like banana bunchy top virus and banana streak virus, and then pests like nematodes and weevils. And the major strain about that is, you will see that many of these pathogens and pests are coexisting, not in the same country, but also most of the time on the same field. So that's why the farmers are really, really worried how to increase the banana production, because if you see on this map, you can see the different colored dots that are different pest and pathogen, and in the same place you can see several of them. So that means that those pests and pathogens are coexisting in those countries. So the genetic improvement of banana in my group actually, or I should say at IITA, basically we apply different approaches, starting from selection of resistant varieties, conventional breeding, then genetic modification using transgenic, and as well as genome editing. And because the genetic improvement of banana holds great prospects for improving food security, and there is critical need because of the presence of so many diseases and pests to develop improved banana varieties with broad spectrum resistance to various diseases and pests. So I will give you a few examples what we are working on. So first example is the banana Xanthomonas wilt, and in short is called BXW, is a bacterial disease caused by Xanthomonas campestris pathovar mucosurum. This is the most devastating disease in East Africa, in the Great Lakes region, which is also the major banana producing area. So this disease was first reported in Ethiopia long back, more than 50 years back, on crop called insect, which is closely related to bananas, and after that it was reported in on bananas. But from Ethiopia, the pathogen actually moved to other countries. It was first reported in Uganda in 2001. And after that, pretty soon it was reported in other countries like Kenya, Rwanda, Burundi, Tanzania, and DR Congo. And then the losses because of this disease is pretty high. It can range from 40% to 80% depending upon the countries. Like in Kenya is 40%, and then in DRC it was reported about 83% because of the losses because of the BXW. The symptom of this disease starts with the wilting. So it starts wilting of the leaves, premature ripening of the fruits, but very soon the disease, the pathogen is spread, and then the fields look like this, so the complete wilting of the plant, rotting of the fruits, and even though some of the plants which, the fruits which looks okay from outside, but then they are rotten and they have the yellow ooze because of the bacteria. The disease is spread mainly through the insect vector. So the insects actually takes the pathogen into the healthy plant. And what happens in bananas, these flowers, so the bracts falls off, leaving the moist cushion, and insects like feeding on this sap here, and that's how they carry the bacteria from one plant to another. But besides the insects, the pathogen also spread to infected planting material, the contaminated farming tools, and as well as the banana traded banana products, because as you can see here, the bananas are wrapped in the and then some bacteria might already present in these sheets, so that's how it can go a long distance as well. All the cultivated varieties tested so far are found to be susceptible, even though there is significant difference in the susceptibility among the various cultivar. There is a wild type banana called Musa balbisiana, which is found to be resistant. And recently there are Musa acuminata subspecies Zebrina and Benkesai also found to be tolerant. But there are some of the varieties of banana which has this persistent bract, And because the insect cannot now, this with the persistent bracts, the bracts doesn't fall off. So, the insect cannot feed on the moist cushion, and that's how actually these type of varieties are escaping the insect mediated transmission, but they don't have the cell mediated resistance. So that means, if the contaminated farming tools are used, then these plants still get infected. And overall economic losses are estimated between 2 to 8 billion over a decade because of this disease. And as I mentioned, that all varieties are susceptible and they result in the absolute crop loss, because as you see the figure, the bottom picture, that's how the field looks like in few months' time once the disease is there. So resistance has been the best and the most cost-effective method of managing diseases. So we were applying the transgenic approach, and we have made quite advancement using 2 different genes, the plant ferredoxin-like protein, PFLP, and hypersensitive response assisting protein, HRAP. Both of these genes are defense genes, isolated or cloned from Capsicum annum, and they intensify the hypersensitive response because of through a harpin, which is a protein secreted by the bacterial pathogen. These genes have been shown effective against many bacterial pathogens such as Erwinia, Pseudomonas, Ralstonia, and Xanthomonas, and that's why we started testing these genes in banana. In addition to this, we were also testing some other genes like XC21 and NH1, and as well as the EFR from Arabidopsis. So in our lab, we have very robust transformation system that allow us to develop transgenic products, and we use cell suspension, and we have cell suspension of several varieties. So these are the single cell suspension, and that's a system we use for making that for transformation. So using the same transformation system, we developed the transgenic bananas. Initially we tested these genes separately, the PFLP and HRAP, and then we evaluated the transgenic lines using a rapid assay, which is using the in vitro plantlets, and we artificially inoculated the bacteria into the pseudostem. And the first picture is the control, where the control started showing the symptom in 10 to 12 days and completely wilted within 1 month time. And whereas that some of the transgenic lines didn't show any symptoms, so we did this assay for 60 days, and the lines which didn't show the symptom until 60 days, actually then we have further tested them using the potted plants, and we have inoculated the pathogen in the leaf, the second fully open leaf, where you can see the blue tag. And majority of plants didn't show any symptom because we have already done the first screening, whereas the controls were completely wilted. After that, we try to isolate the bacteria from the 5 centimeter above and the below the point of inoculation, and we couldn't detect any bacteria through the culture or through PCR. But we clearly see the hypersensitive response and necrosis at the point, and so that means it creates a physical barrier where pathogen is quite confined, so it's not moved, and that's why the plants are not showing the symptoms. So we further tested these in the field trials in Uganda in collaboration with NARO, and in Kenya in collaboration with Calro, and we got very good result from the field. Several of our lines showed complete resistance to the bacterial wilt. So this is our transgenic line. You can see there is no symptom on this line, whereas the control plant completely wilted. And so we artificially challenged this plant at the pre-flowering stage, So by the time there was the majority of the control plants actually died before flowering. Some of the plants actually did flower and produce the bunch. So the lower is the control non-transgenic bunch, where you can see the symptom. Only few plants produce the bunch, but our transgenic bunch has no symptom. And then for this work, we used Sukalindisi, the variety which is a dessert type of banana, is a small banana, so that's what you are seeing here. Then we also check the internal symptoms. So the upper panel is the transgenic line, which are not transgenic fruits, and this is the pseudostem, and they are not showing any yellow ooze. And bottom panel is the control non-transgenic plant, where you can see in the fruit the necrosis and also the yellow ooze. In addition to the disease resistance, we also looked at the plant morphology and the yield component, and so we collected the data for the disease resistance for several generations. Actually, in total, we did that for 5 generations, but this graph only shows the 2 generations, the main mother crop and the return crop, and you will see several transgenic events which has both crops have the complete resistance. But also the plant morphology, we didn't see any significant difference between the transgenic and the non-transgenic, and same for the yield component as well. So after that, we did the non-target effect, because that's very important if you are particularly interested in product development. And the expression of PFLP and HRAP genes didn't show any effect on non-target microorganisms in soil. We also tested the biosafety of PFLP and HRAP. So we did potential allergenicity and toxicity using the bioinformatic analysis. Actually, we started this work before advancing the transgenic, and then because every year there are new allergens or toxins are listed, so that's why we are repeating this every 3rd year. Initially, I was doing it myself, but then, you know, in order to build confidence of the regulators we are now, University of Nebraska, Lincoln is actually the one who does this work for us, and we didn't find any convincing evidence to suggest that either of these 2 proteins represent any risk of allergenicity or toxicity. For the bioinformatics, we did the 80-mer sliding window search, but some of the regulators might require for single mer, so we also did with that, and we didn't find any evidence to say that it has any toxicity or allergenicity. The group, the socioeconomic group at IITA also did the potential adoption. So they did the ex ante impact analysis, and whereas so they did the survey, including both the consumers as well as the producer, so they raised this question about the willingness to adapt this GM bananas resistance to BXW, and 65% of the growers says that they will immediately adapt. About 19% said that will adapt, but after seeing the performance of these bananas in neighbors' field, and then only 16% said that, you know, they will not. So which is also not very critical, you know, 16% is small. And we also did the projected adoption rate of GM bananas resistant to BXW for all the 6 countries where this disease is impacting the banana production. And so it will start with the slow pace, like starting with the 20%, but then within few years time it can be adapted at up to 100%. And the adoption rate was the highest in the country where the losses were highest as well. So that's DR Congo. One thing which is very interesting, which I found very interesting actually, was that, you know, for farmers who are affected with this disease are actually, for them, it doesn't matter whether the biosafety regulation is in place or not. What they want is the resistant varieties, because DR Congo doesn't have the regulatory framework for growing these bananas, but on our survey, that was the country which has the highest adoption rate. So currently we are on the product development status. So for the product, as you know, that the single gene based resistance break down very easily. So we have stacked the two genes together, and now we are at even characterization and selection. So we have done two things. The first thing, the proof of concept, was using the easy to transform variety, which was Succulent DZ, but later on the stacked gene constructs are transferred to more farmer preferred varieties, and now we are at the even characterization and selection, and now we will be advancing these to the multi-location, and in parallel we are also already started doing some regulatory science where we are checking for the biosafety and also the even characterization, and then we will after that we will move to the regulatory trial. As I mentioned that bananas has lot of diseases and pest pathogens, so we also did some work on the nematodes, because the nematodes can affect bananas. The losses can be up to 57 to 70% depending upon the varieties. The plantains, which are grown more in West Africa, are actually more prone for the losses because when the nematodes makes the plant weaken, and then in West Africa there are a lot of storm, and because of that the plants topple. So there are some limited resources of nematode resistance or tolerance in Musa gene pool, but then that's against the one species of nematode, that's Rhedophilus similis, and also that it still needs to be combined with the consumer acceptable traits in the farmer preferred varieties. But when you go to the field, actually it's not only one nematode which is present, is several species occur in the same soil. And so that's why we were collaborating with University of and we were using the gene stacking for actually developing the broad spectrum resistance against several species of nematodes. So we stacked two different technologies. One is the cystatin from maize, which is a cysteine inhibitor, and another is a synthetic peptide which acts as repellent. And so we have stacked two of them together and developed the plantains. And so after developing the transgenic plantain, they were molecular characterized, and after that tested under the glass house, and then later on in the field trials in collaboration with NARO in Uganda. And then we found several events which had shown quite high resistance to nematodes. We are also working on a project where we are developing RNAi technology for controlling the banana bunchy top virus. Banana bunchy top virus is actually transmitted through aphid. So we were trying to develop the proof of concept for both the virus as well as the aphid. And our intention is that once these technologies are developed, then we can actually stack them together for durable resistance against the banana bunchy top virus. So moving to cassava, cassava is another very important food security crop for resource limited or smallholder farmers. And despite its importance, actually it also has a lot of constraint, and therefore the improvement of cassava is also pretty much needed. So we have developed the cassava transformation system at IITA, and we can transform several varieties of cassava. And for cassava, our target is basically the cassava brown streak disease, which is a viral disease. It is caused by two viral species, cassava brown streak virus and Ugandan strain cassava brown streak virus. And it is a pandemic throughout the East African region. And currently there are some tolerant varieties, but there is no variety with a complete resistance. So we are developing the transgenic cassava using RNAi technology against the virus, and again also against the whitefly, because the whitefly is the one which transmits the cassava brown streak virus. And this project is in collaboration with Venganza in US and USDA. Yam. So yam is a Dioscorea species. Many people confuse yam with sweet potato, but it is not. Is a Dioscorea, and it is also polyploid and vegetatively propagated crop. There are a lot of species of yam, but then only few species are cultivated. It is a food security crop which and provide income to over 300 million people, particularly in West Africa, because West Africa is the major yam producing region. IITA has a very strong breeding program for yam, but then there are a lot of challenges which breeders are facing, and maybe through biotechnology, maybe we can complement some of them. So we have developed the yam regeneration through somatic embryogenesis, and also the transformation protocols. Yeah, so this is the transformation. Next slide, please. Okay, so now I will switch my gears to the genome editing. So as you know that genome editing for crop improvement is now becoming quite popular because it is precise and very effective. But you know, genome editing is not new. Nature has been editing the genome for a very long time, creating variation. And in old days that, you know, farmers were selecting these variation for cultivation. But in 20th century, the mutations were accelerated through chemicals and radiation. So, you know, there was the chemical induced mutations, which was in 1944 the first time reported, but then the radiation also in 1927, and later on the more precise gene targeted technologies were developed, like zinc finger nucleases, meganucleases, and more recently then the CRISPR became the most popular genome editing. And so you know that this last year actually this CRISPR technology got a Nobel Prize. But even within the CRISPR, actually, it is advancing quite fast, you know, every other time of gene editing, because they have the implication on its. Sorry, I think it was disconnected. Now I'm back. So in genome editing, the site-directed nucleases, it creates the double-stranded break in the double-stranded, and then after that, you know, the repair can happen in two ways, using the non-homologous and joining when there is no donor template, and in this case, it creates the random targeted random mutation, but it is normally small indels. And this caused the gene silencing, gene knockout, or the non-functional protein, and this is called SDN1. In the SDN, and then when it is the donor templates is there, so homologous recombination. In that case, there is either correction using the donor DNA and with a small, so it's a introduction of the mutation at the target site, and that's the SDN2. And then in SDN3, which allows the introduction of the full gene, which can be used for the allele replacement or even the gene insertion, but at the target site. So this is SDN3. And it is very important that you understand the type of genome editing, because it has the potential discrepancies in the regulatory approaches. So SDN1 and SDN2 types, where is only the small indels or mutation, but there is no foreign gene integration. In this case, they are very similar to the mutation obtained through the chemical mutagenesis or irradiation or the spontaneous natural mutation. And in several countries, this type of gene-edited products are not regulated, similar to the transgenic. Whereas the SDN3 type, where there is a full gene insertion, it can be cisgenic or transgenic. So application of genome editing is quite broad. It can be used for the yield improvement, like grain weight, grain size, numbers, but also for the biotic stresses, resistance to bacteria, virus, fungi, abiotic stresses like drought, heat and cold, herbicide tolerance, also for the functional genomics, which in our lab we are doing quite a lot on gene discovery and functional genomics using this, and the nutrition enhancement like proteins, vitamins, oil, starch, and sugar. We started genome editing of banana quite long back in 2015, and our lab has the first approval from the National Biosafety Authority in Kenya for conducting the genome editing, and particularly for the virus resistance. And so we developed the protocol for genome editing targeting the PDS gene. So if this one is, we used it as a visual marker. So if the gene is functional, then the plants are green. But if you make this gene non-functional, then you get the albino plants like you see in this picture. So then we detected the targeted mutations with the sequencing of the amplicons from this. And developing, then we have the proof of protocol, a proof of concept for knockout of banana streak virus. I think I'll present that later. And now we are working on the bacteria wilt and the fusarium wilt, and we are planning to start testing some of these ones under the field conditions soon. So banana streak virus, it is a double-stranded DNA banana virus which integrates mainly in the B genome of the host plant. So banana has two genomes, A genome and the B genome, and it is a triploid. So so it can be triple A, or it can be AAB or ABB. So in plantains, which is AAB, this streak virus is a big problem. So banana streak virus is a monopartite. It has a monopartite genome with 3 open reading frames. Open reading frame 1 and 2 is comparatively small, and they are involved in the virion, either virions or virion assembly. But open reading frame 3 is a big open reading frame, and it encodes a polyprotein which is cleaved post-translationally, and then it makes the functional proteins like movement protein, coat protein, as protease, reverse transcriptase, and RNase H. But then it integrates into the B genome in the multiple copies as direct or inverted tandem repeat, but it always integrates at a single locus in the B genome. And when it is integrated, in the integrated form, the plants doesn't show any symptom. But under the stress condition, this integrated form, the episomal form, and that's when the plant start showing the symptoms. And so this is becoming a big challenge for the breeding program, and as well as the dissemination of the plantain hybrids, because tissue culture is a plant considered tissue culture as a stress, and it normally activates the integrated virus. And another thing is the breeders cannot use the diploid Musa balbisiana, which is a wild type banana, but then because it has Musa balbisiana is BB, so it has 2 B genome, but most of the time, the Musa balbisiana has resistance to several pathogens. But because of this streak virus, breeders don't use this in the breeding program. So the we are using for inactivating the endogenous integrated banana streak virus is that we are using the multiple guide RNA targeting all the 3 open reading frames. And open reading frame 1 and 2, our intention is that if we can simultaneously target 2 sites, and then so it can be the large deletion in the two target sites. And then in the open reading frame 3, we target the aspartic protease. And then if we make the aspartic protease non-functional, we can actually make all other genes non-functional, because the aspartic protease is the one which cleaves to make the post-transcriptionally functional proteins. So with including these 2 approaches, we can make the virus inactivation. So this what we did for this one, we have actually we were working on one of the plantain cultivar called Gonja manjaya, which is triploid AAB, and we collected the planting material from the field, which is showing no symptom under the field condition, and we introduced them to the tissue culture. And then after that, we have characterized this to find out what type of pattern of integration. So we didn't see any episomal form of BSV, and which was very obvious because there was no symptom in these plants. We only find out that, you know, it has the integrated endogenous, and we did characterization using different primers and as well southern to confirm this pattern. So based on strategy which I presented, so we have 3 guide RNA. We use the plasmid mediated plasmid-based delivery of these guides using agrobacterium transformation into the cell suspension. We generated so these were transgenic events because, you know, we use the plasmid, and so we confirm them through for the presence of the Cas9, and after that we did the band shift analysis. So this is the PCR. We designed the primers flanking the target site in open reading frame 1 and 2, and we were expecting about 198 base pair deletion, and so that's why we saw the band shift in some of the lines. Then after that we confirmed that by sequencing, and we saw the deletion, the big deletion of 198 base pairs in several of these, the mutants. For the open reading frame 3, the target site, we saw only small indels because there was only a single guide. And then, so the plants, we potted them in the glass house, and the plants looks quite normal compared to the non-transgenic or non-mutant wild-type plants. So then after that, we phenotype these plants. So basically, one of the stress is also drought. So, you know, we gave them the water stress. We didn't water these plants for several weeks. So this is the first picture is of the control plant, and you can see very clear yellow streak of the banana streak virus in the control, but then the mutants didn't show any symptom. These the 3 mutants, but the 2 showed some mild symptoms. So we were wondering what was happening. So the 2 symptomatic events had indels only in open reading frame 1 and 2, and they didn't have any mutation. Suggests that, you know, open reading frame 3 plays a crucial role in symptom development. And as you remember, I said that it encodes the polyprotein, which is cleaved post-transcriptionally by aspartic protease into the functional protein. And so we tried to make the aspartic protease non-functional. So indirectly we were making all the proteins non-functional, and that's why this virus was not activated once the lines which has the the mutation in open reading frame 3. And we further tested the presence of the virus using the PCR and QRT-PCR. So currently, what we are doing is, because we want the product which is non-transgenic, no foreign DNA integration in it, so we are trying to improve the parents which can be used in the breeding program. So we are using that approach, knocking out the endogenous streak virus. So we are using 2 different parents. One is the 4x hybrid, and then the second is the 2x improved diploid. And then once we have the mutants for these, and when we have detected, then these will be crossed through our breeding program, and then we will get this improved triploid, which will be plantain hybrid, which will not have the streak virus. So this variety will have the inactivated streak virus. Now what we are also doing, we are also focusing on the disease resistance for bacterial disease as well as the fungal disease. So you remember I mentioned that Musa balbisiana is resistant to banana bacterial wilt. Genes in Musa balbisiana, and we are trying to overexpress these genes, these defense genes, using CRISPR activation, as well as through the transgenic approach, and we are knocking down the susceptibility genes in the susceptible varieties in order to create the resistant variety. So we are using the information from Musa balbisiana to develop the bacterial wilt resistant banana. So that will be our second generation product for the bacterial wilt resistance. So we did the transcriptomic, and we compared the resistant variety, which is the Musa balbisiana, with the susceptible variety, and then based on that we identified several susceptibility genes as well as the transporter genes and transcription factors which are involved as negative regulator of defense pathway, and that's how we are targeting those genes for knocking out. So one of the gene which we are targeting for developing resistance against bacterial wilt is the DMR6, which is a downy mildew resistant 6 ortholog in banana. So we did the phylogeny of the 2-oxoglutarate iron dependent oxygenase, and because DMR6 comes under this gene family, and then you see the blue highlight, that's the clade with for the DMR, where the DMR6 belongs, based on the Arabidopsis thaliana, and we found 7 orthologs in Musa acuminata on the A genome, and 2 orthologs on the B genome in the Musa balbisiana. And so what we are now trying to do, we are not knocking out all the orthologs, because if we do that, then it will definitely impact the plant yield and the plant growth. So we have only identified 1 ortholog which we are trying to knock out. So we have now identified which exon. So we are actually on the exon 4. We have designed the guide. So what DMR6 overexpressed during the pathogen infection, and this gene is actually activated during the host pathogen interaction and suppress the plant immunity. So we have done QRT-PCR after challenging the plants with the bacterial pathogen, and in Musa balbisiana we didn't see any differential expression, but in the susceptible variety Succulentaceae, we saw 60-fold higher expression of DMR6 ortholog, which we are trying to knock out now. So we have developed the Sucalindese mutants by knocking out the ortholog of DMR6. So we have regenerated the plant. So we again use the two guides here. So this is the gel image showing the band shift because we use the two guides. So if the Cas9 cuts simultaneously at the two targets, then we see the big deletion. And we, in some of the lines we saw that one, and then so we have further confirmed the targeted mutations using the sequencing. And then we have evaluated these ones for the disease resistance using the rapid bioassay, the in vitro, as well as we saw the enhanced resistance to the banana Xanthomonas wilt. Because DMR6 has been reported to also impact the plant growth. So we also, in the glass house, we also collected data on the plant growth. And so far, we have not seen any negative impact on the plant growth of the mutants for the DMR6 for both for the plant height as well as the total leaf area, but we still need to confirm this result under the field condition because so far we have the result only from the glass house. We are also working on Yam genome editing because the reference genome of both Dioscorea rotundata and Dioscorea lata are available. So we have established a genome editing of Yam, and this project we are working in collaboration with Iowa State University and University of Michigan, and now we have the system in place. And now we are looking for funds so that we can start developing the genome edited products for Yam, targeting the Yam viruses and anthracnose disease. So there are some biosafety concern with the genome editing, but there are two concerns. One is the unwanted genetic changes in plants due to off-target mutation. But off-target mutations can be minimized. It can be done by designing the guide RNA strategy. So what we do is we select the guide RNA which has the high efficiency for targeted mutation and then the very low potential for the off target. So that's how we start with. And also, if you use either nucleoprotein delivery or using the inducible CRISPR-Cas, so that the CRISPR is not activated the full time, then you can also reduce the off-target effects. It means with that type of approach, the CRISPR-Cas has created the mutation and then they are degraded, and in that case the chances of the off-target mutations are really minimized. The second concern is the transgene integration, and then that can be handled either using the DNA-free genome editing where there is no foreign DNA integration because the delivery is in the form of pre-assembled. So there is this slide I want to show like the global review of legislation for genome editing. So the countries which are highlighted in green, these are countries where the SDN1 and SDN2 type of gene edited products where there is no foreign gene integration are not regulated as GMOs. Just talk, without slides.
Yeah, that's okay.
Yeah. So the countries I was talking about is like US, Canada, Colombia, Brazil, Argentina, Chile, Australia, Japan, Israel. These are the countries where the gene edited products, if there is no foreign gene integrated, are not regulated as GMOs. But then there are several countries where currently the guidelines are under discussion. For example, in Kenya, Nigeria, also in India, Bangladesh, Philippines, Indonesia, Paraguay, Uruguay. So these are the countries. Also here there is the discussion going on that if there is no foreign DNA integration, then the products will not be considered as GM. So which is really good news, is apart from Europe and New Zealand, where currently the gene edited crops are regulated as GM. So currently that's the legislation globally about these GM crops. Yeah, so I think that was my last slide, and then so few key take-home messages are like application of biotechnological tools has potential to transform agriculture. Genome editing tools are becoming popular molecular tools of choice for functional genomics as well as crop improvement, particularly the CRISPR. And CRISPR is rapidly increasing, and it's becoming the most popular genome engineering approach due to its simplicity. It's pretty simple, you know, any lab which has the transformation ability and some bioinformatics, that then they can go ahead and do the CRISPR. So it's simple, efficient, it's specific, multiplexing, and it's easy to adapt. And then genome edited crops with no foreign gene integration are not regulated in several countries. There are availability of reference genome sequences and transformation system for several RTB crops, which make them suitable candidates for genome editing. And the application of CRISPR-Cas based genome editing has been demonstrated for some of these RTB crops, particularly banana and cassava. So in the end, I would like to acknowledge my team, my transformation group, the bioinformatics expert, virologist, and also our partners, NARO, CALRO, University of Leeds in UK, University of California, Queensland University of Technology in Australia, Iowa State University, Academia Sinica, Taiwan, and then the financial supports from several donors like Bill and Melinda Gates Foundation, CGIAR Research Program, DFID, Gatsby Charitable Foundation, USAID, and National Science Foundation. Thank you very much, and sorry for the technical hitches.